today I'm going to be going over the physiological anatomy of the skeletal muscle. I'm going to be numbering the diagram with the corresponding numbers to the right. So first I'm going to start with the muscle fiber. The muscle fiber is a multinucleated cylindrical muscle cell. And these individual muscle fibers are going to be separated by the endomysium, which is here, number 2. So that is a connective tissue that's going to go in between the fibers. These bundle of fibers, or fascicles, is another word for them. That's what a bundle is going to be, a fascicle, is going to be wrapped in perimysium. And perimysium is a connective tissue that holds together that bundle of muscle fibers, or fascicle. We're also going to have blood vessels in the skeletal muscle. So blood vessels are going to be aid in muscle regeneration and muscle development. They're going to provide oxygen and nutrients to the muscles. And for instance, if you're exercising, you're going to have an increased flow of an increased blood flow to meet those oxygen requirements for the skeletal muscle. So that's just one example. Then we're going to have this whole muscle surrounded by epimysium, which is a dense connective tissue that really defines the volume of that muscle. And attaching the muscle to the bone is we're going to have the tendon. And the way that I like to think about the tendon is a lever that moves the attached bone when that muscle is contracting or relaxing. So now let's take this myofibril and zoom in a little bit to see what else is going on inside of here. So on the outside of that muscle fiber, we're going to have the sarcolemma. And the sarcolemma is basically the plasma membrane of the muscle fiber. And just like any other cell membrane, it's going to regulate the exit and entry of materials from that cell. Then we're going to have the mitochondria, which is going to be converting those nutrients to ATP. Because of the high energy requirements of the skeletal muscle, we're going to have many mitochondria. And then we're going to have the myofibril, which is the organelle responsible for muscle contraction. It's going to be made out of a bundle of protein filaments, and there's going to be many chains of these myofibrils within the muscle fiber. And like I said before, the muscle fiber is multinucleated. The reason for this is that among vertebrates, muscle fibers are actually the biggest cells. So for each nucleus is going to regulate the surrounding sarcoplasm. Now, here we have the striated appearance of the muscle fiber. This light A band is going to be made up of thin actin filaments, which is why it's going to be getting a little bit lighter. But the dark A band is going to be made out of, it does have the thin actin, but it also has the thick myosin filaments, which is why it's going to be a little bit more pigmented. So here we're going to have the dark A band. Now let's take this myofibril and zoom in a little bit more. So like I said before, we're going to have these thinner filaments within the myofibril, and then we're going to have the thicker filaments, thick myosin. So thin actin, thick myosin. Now, we're also going to have in here the sarcomere, which is the main contractile unit of the muscle fiber. And it's going to have actin and myosin in it. And those filaments are going to be responsible for muscular contraction. So sarcomere is going to be this region right here. This region right here, these arrows. That's going to be 3. This region right here, which I was just pointing to, is the A band. The same A band that we mentioned before when I was going over the muscle fiber. And as you can see, I've numbered it the same. So the dark A band is going to be number 5. Number 4 is going to be the I band, which is only going to be the thin actin filaments. Number 5 is actin and myosin. Number 4, the I band, is going to be just the actin filaments. So that's going to be number 4. And also over here is only actin filaments. The H zone is going to be just the thick myosin filaments. There's going to be no overlap of the actin with the myosin. It's just going to be the myosin. So that's going to be the H zone. And then over here, we're going to see the Z disc, which if you scroll down, you see that's number 7. And the Z disc is at the end of the sarcomere, and it binds the filaments and really stabilizes that sarcomere. The distance of the Z disc between each other is going to shorten when we're doing contraction. And then lastly, we're going to have the M line. And the M line is where it's going to be in the middle of the H zone, and it's where the accessory proteins are going to bind to myosin.